move to this last presentation. This is complementary of what Elka mentioned on the lecture. So he introduced the homology. He showed the link between Laplacian operator, and he is mostly dealing with let's say one structures of the geometry. So we can look at this geometry in a particular way. Now, when you like to study, the relationship between several, phenomena. Let's say one phenomena talking about for instance, we are dealing with the brain, how the brain, from the anatomical point of view, connect between different areas. We all say this is geometry. We can represent this in a certain way, and homology can look at it. But if you come and say, "Oh, guys, I know this," or a little bit, but how the metabolism goes between different areas, oh, this is another question. Okay, well, we can do the same, but if you ask me, oh, what are the neurons doing from the electrical point of view? This is another question. And if you say, oh, now I am looking at the genome of the different area and how is the structures of the genome is distributed from the geometry point of view. So you are adding different knowledge, different skills, but the fundamental question is how you can bring all this sandwich of a geometrical representation to extract some features who talk and say the geometry for all of this is this, and we can have some invariant who can explain how the brain behave. Okay, so this is what Elka generalized, like a cross-homology. This is the link between several geometry, and he generalized this at the level of the Laplacian, because homology Laplacian. So what about if I move from one homology to another one? So this is a cross-Laplacian, and this is another notion who generalized this multi-layer data analysis. So Elka, thank you for this presentation, and you have 45 minutes.
Okay.
Sure. Thank you.
Yeah, thank you, Habib. Oui. Can you see my mouse?
Yes.
Okay. Okay, so hi again. So I gave you all the basic notions of simplicial homology and Hodge-Laplacian theory. So now I'm gonna bring you into some again higher dimensional extension of everything I've said about, and this is called cross-homology. So what's wrong? Oh, okay. So again, there is a limitation with graph theory and simplicial complexes, and the limitation is the following: is that in real world data, there are different interactions that are involved. For example, for the brain, obviously, they are connected by fibers, but they are also connected by functions, and even structural connectivity. If you go in the macro scale, So you have different connections between neurons, gap junctions or chemical, etc., etc. So it can be also by external connections via neurotransmitter, etc., etc. So there is the need to set some mathematical formalism for all of that. And the formalism that has been used so far is called multilayer networks. And it is a very straightforward generalization of a network, meaning that you have different layers of networks. For example, in real world, you can have, if you take the urban transportation system, you have different way to move from one point to another via metro, bus, bicycles, etc., etc. So in that case, you have multiplex because you have nothing between the layers. But in some cases, you have also connections between the layers. For example, let's say, again, to go back to the social, to society. So let's say every layer, each layer here represents a different kind of interactions between people. Let's say this is friendship, the V1, and V2, let's say this is professional relationships, and V3 is kinship relationship, family. And between, you put anything you want. For example, between V1 and V2, economical ties between people. If, for example, you are connected via a company, ownership, for example, that's different from professional, I mean, a traditional professional relationship, and V2 to V3 and another relationship. So now you want to understand the whole structure, to analyze the whole structure in a unified framework. You might ask why? Why don't I just consider the whole structure as one network and apply the usual graph theoretic measures to analyze my network? Well, the problem is that if you do that, you lose the specificity of the connections. And what if you lose the specificity of the connections? Then you lose insights. You might dismiss some structures. For example, if your aim is, for example, to understand how a virus spreads in a social group, then you really need to understand how people connect to each other and how often do they connect. I said, in that case, you need to understand all those parameters, all those, you need to take into account all those different type of interactions. And you cannot neither study each layer separately and then compare them. That doesn't make any sense. I mean, okay, it depends on the questions you ask, of course, you want to address. But anyway, so the idea is to study all the whole structure within a unified framework. But and these ideas have been used in different area in neuroscience specifically. So, for example, here this is the connectome of the C. elegans. So I don't know how much familiar you are with the C. elegans. The C. elegans remains, as of today, the only animal whose connectome is completely known. So it has only 302 neurons. And yeah, that's sad. But despite this very small number of neurons, it has been shown that it is able to actually to do some difficult tasks. For example, it can learn. So it has the ability to learn from past experiences. For example, so one experience was to put the worm on a table and to tap like this. So they actually noticed that first the worm would actually go faster, try to move from the danger. But if you continue, it understands that there's no danger actually, and it goes back to the its normal speed. So which means that actually it can learn from past experience. So meaning that in fact it's not really the number of neurons that brings to complexity, but the way the topology of the network itself. So, and the topology here is defined by different kind of networks. Here you have the synaptic, and you have the electrical connections, and here you have different networks of neurotransmitters, monoamine and neuropeptides. So in this is a very interesting paper where they studied even these neurotransmitters via different genes, and they consider all those layers and they put them together and they try to understand the whole structure. Well, actually here, no, that's the drawback of this paper, is that they didn't study the whole structure as a whole, but they study each layer separately, and then they compare using the traditional graph theoretic measures, and they compare them together, and they compare each layer with the random network defined by the same number of nodes and the same degree distribution. So that's what they did. But it's still a very interesting. I mean, it's new. I mean, it's new. There are new tools. So obviously there are some. Yeah. Anyway, so the problem here again is the same I mentioned for graph theory, for the use of graph theory in real data, is that very often real data, empirical data, involve more, even when multi-layer network data involve more, involve interactions involving more than two nodes. It's the same for the society as well. I mean, three groups, three people that form the same group is different than three people with three relationships. That's different. And you can also map the same social network to another social network. For example, professional networks, you will have the same observations, and you can have the same different kind and different dimensional interactions between the two layers as well. So this is the limitations here. So we address these limitations by generalizing the idea of simplicial complexes to multi-layer simplicial complexes. So this is an example of that kind of structure. You would have horizontal simplicial complexes. So they are simplicial complexes in their own right, and they are linked together by other simplicial complexes. So it's different layer of simplicial complexes and interlayer simplicial complexes between V1, V2, and also between V1, V3, and between V2, V3. So they are so each pair of layers is interconnected by different kind of simplicial complexes. So now the question is, how do we study the structure and the topology of such a structure without separate each subsystem from each other? So we did that by, so this is a paper we've just published. So we developed this theory of cross-homology and higher dimensional cross-Laplacian operators. So the same idea, but in different dimensions. Except that now, this is a a very simple example of such structure with different with two layers. So we call this a cross-simplicial bicomplex, because we have two complexes. So basically, you have the same structure. You have, for example, here, this is a cycle, this is a triangle, two triangles here, and cross triangles. You have different kind of cross triangles. A triangle, let's call this one the green one, bottom cross triangles, because they are formed by one node from the top layer and an edge in the bottom layer. This is different from the light blue triangles. This one, V12, V01, V11, for example. It's different. Okay. So now the goal is to study this topology and also to have interpretation, to see actually what is a cavity in such a structure. What holes look like. Okay, we know what is a hole in a horizontal simplicial complex. We can use the same method, the same simplicial homology here to study each layer, of course. But the question now is the interlayer structure. Okay, so you will see that in fact this configuration, so V14, V24, V16. You see here, this configuration is an actual hole. This is an interlayer hole. But if you flatten the structure, you wouldn't see that, because the three nodes are connected, so you wouldn't see that hole. And the same thing with this thing here. So in a flat network, that would be a cycle. Yes?
What is the interpretation of that hole in a tangible?
Interpretation. Okay, so I will make an attempt to give an interpretation when it comes to understand the multilayer network formed by functional connectivity and anatomical connectivity. So that would be, for example, how the functions emerge through the anatomical connector. Okay, so you have already the structure, and you want to see how the function, how the function draws its topology across the underlying anatomical structure. So it would be, for example, let's say this is anatomy and this is function. Let's say these structures are functions. That would be, well, a functional integration, for example, between two anatomically connected networks, two fully connected networks, but separated by functions. They're, well, anatomically separated, but functionally connected. So it would be a functional hub, for example. But it would actually give make a difference between a functional hub between two separated regions that are not connected to any other, well, it doesn't exist, but let's say they exist, and another functional hub that connects two networks, for example, anatomical networks. Now, what do we say about that? That's another question. Okay. Okay, so so this is, like I said, this is what, so you have different kind of cavities, interlayer cavities. For example, you have this one, but also you have this configuration here, this cycle formed by one node, one top node, and a path in the bottom node, in the bottom layer. That's also an interlayer cavity. Okay, so here is a sketch of the formalism behind this idea. I won't go deep in this stuff because you already get I think from this you already get the idea. So a cross-simplicial bicomplex, I will start by the building block bicomplex, just considering only two layers. So let's call the top layer the vertices just by a coherence in the notation x0 minus 1. Just a notation x0 because I'm considering only the nodes from the top layer and nothing. So I'm considering only these nodes separately, forgetting the other layer. And then I do the same for the bottom layer, x minus 1 is 0. So the minus 1 is for homological notation, for coherent, etc. You can put whatever, anything you want, but I want it to be consistent here. So we have two simplicial complexes, x0 minus 1 and x minus 1, 0, and simplexes between them. They're linked with simplexes. So let's call the vertices V1 and the top vertices and the bottom vertices V2. So now the bicomplex consists of objects of this form. You have some number of nodes from the top layer and some number of nodes in the bottom layer, and all of them form the cross simplex. So in fact, if you forget all the bottom nodes, you should have, and you consider all the top nodes that are inside that space, they should form a simplex in its own right in the horizontal complex. And the same observation for the bottom layer. So that's if you think about graph, you just consider if you have two graphs and links between the two graphs, you would have clicks on the top and clicks in the bottom, and all the nodes are connected. They're all pairwise connected. Okay, and this is a KL cross simplex. So we do the same thing. So we call the XKL all the KL cross simplices. And here is a geometric representation of these objects. So this is simple. You have a triangle, edge, and a node. You have an edge, cross edge, and you have a cross triangle. Again, you have two different kinds of triangles. This is a top triangle. So a triangle formed by one node from the bottom layer and an edge from the top layer. But you also can have triangle like this, like I said before. And this is a cross tetrahedron. Here you have three kinds of cross tetrahedron. You can have two connected nodes on the top, two connected nodes on the bottom, and the four they are all pairwise connected. That would be tetrahedron. But you can also have one node in the top layer and a triangle in the bottom, and edges between all of them. Or the inverse, the triangle could also be on the top layer, etc., etc. So this is a geometric representation just to give you an intuition about these objects. So now you don't need this, but just to give you that. So this is exactly what we did for simplicial homology. In fact, what we do is that you can go from higher cross simplices to lower cross simplices in different way. Let's go back here. For example, if you take this cross edge, you can remove this thing, this node, the top node, and just keep the bottom node, and in that case, you are in C0, the C0 of the bottom layer. But you can also do the same thing for the bottom layer. Remove the bottom layer, the bottom node, and keep the top node. In that case, you go to the C0, you land in the C0 of the top layer graph. Okay, you can do the same for triangles, remove this, and you get an edge, etc., etc. So this is the idea here. So which means that what you end up having is two homology theory. It doesn't mean the homology of each layer, but just two homology that will give you different point of view of your structure, okay, without cutting them. So, and this is exactly what. So the heart here means that you remove one node. So you go from this to 1 degree lower in the left, so in the top layer, by removing one node. You do the same here by removing one node from the bottom layer. So you do exactly this. So it's really straightforward here. And now you define the same boundary maps, the same technique, because you take the vector spaces generated by these cross simplices, and you define these boundary maps. So you get two boundary maps and two homology. So this is an example, just an example of computation of these boundary maps. And then the homology groups. So two homology group now. Now the homology group are indexed by a pair of indices, because you need one index corresponding to the top layer and another index corresponding to the bottom layer. Okay, so the same thing, but you have now two groups, which will give you two kind of Betti numbers. And these are the Betti numbers. And again, if you consider, so this homology also allows you to study the homology of both complexes separately, but at the same time, the whole structure at once. So again, the computations are more practical when using the corresponding Laplacian. So I define this Laplacian, so two, because you have two homology group, two homology theory, then you need two Laplacians in order to compute them. So I call them the top cross-Laplacian and the bottom cross-Laplacian. We prove the same results here. So the spectrum of each Laplacian actually capture the extent to which higher structure from one layer is connected to lower structures from the other layer. So that's exactly the concrete interpretation of this homology and these Laplacians, in fact. So for example, by higher structure, I mean how edges are connected to nodes from the other layer, how triangles are connected to edges, and how tetrahedrons are connected to triangles, etc., etc. And you do the same thing. So in a way, in a sense, it's a way of looking the other layer through the lens of one layer. For example, if you, like I said, if you're studying functions and anatomy, you want to see anatomy through the lens of functions. That's different from looking at the functions through the lens of anatomy. Okay, so the first viewpoint would mean how the function maps on anatomy, but the other way would be how anatomy defines or determines the functional connectivity. And so there are different way. So in fact, in order to so in our paper, we illustrate all the notions we introduced with a very simple example based on airports and airlines, because it's a very simple example in which everybody can rely on. So we used data sets of all the European airports and different airlines. So two airports are connected. So each layer corresponds to an airline company, let's say Air France, and we take all the airports that are connected by Air France. So two airports are connected if Air France serve those. So there is a direct flight between them by Air France. So, and you would ask, okay, so what is the most efficient way to go from Air France network to the Lufthansa network for some reasons? So you have tickets of Air France, but you want to connect with Lufthansa, and you want to, what is the efficient way? In that case, we show that with using our technique, it gives you the exact airport from which that allows you to go from the Air France network, for example, and connect with Lufthansa or any other company. So homology does that. This cross-homology does that, but the cross-Laplacians gives actually better results, and really powerful actually, because it gives exact. I mean, very few number of airports, so you don't have many choices in that case. So so the thing is, and in that example, the idea is the following. So we have the cross-Laplacians. It is they are linked to the cross-homology via the zero eigenvalues. So here's the one layer. In the case of airports, for example, in that case, you have the same nodes on both layers, right? In that case, what we did is to consider the interlayer connections as, for example, if this is Air France and this is Lufthansa, for example, okay, or let's take what is more interesting to us. This is functional. So, and this is structure. But you will see here what we use actually is not the correlation, but partial correlation. Okay. The reason for using partial connections is that partial connections gives you actually the anatomy and functions. So most often, if two regions are partial correlation connected, then they are also anatomically connected. It means that the anatomical connections that are functionally active, for example. So what we did was to take all these connections and put them here. For example, so have these connections. Here we have the anatomical connections. let's say. Okay. And now what we're doing now is to look at, again, the structure through the lens of functions. So the cross-homology group, H00, this is the one that interests us because this is exactly what gives you the cavity I showed you. So for example, this is a cavity, it will be seen by H00, the bottom H00. Okay, and this is also a cavity, it will be seen by H00. So, and the cross-Laplacian as well, and you look at the spectrum of this. So what is this this operator? This is a matrix that is defined on the cross edges, meaning the indices of this matrix are the cross edges. Okay, but let's say this is region R1, this is region R2, R3, R4, R5, let's say. Okay, so you will have an entity between R1, R2, and R2, R1, R4, R5, etc., etc. So the eigenvectors now will be vectors indexed on the same elements, so the same cross edges. Okay, so we looked at the whole spectrum of this, and for every eigenvalue of this operator, we look at the eigenvector corresponding to this eigenvalue. Again, the eigenvector will be a vector indexed on these cross edges. And then we consider all the nodes, all the top nodes on those on the cross edges with non-zero elements in those eigenvectors. Okay, so that will be hubs, and we call them cross hubs. So harmonic for the zero eigenvalue, we consider the hub. So for example, here in this example, so this node will be a cross hub. This node will be a cross hub, and this one as well. This is a cross hub, because they will appear, all these cross edges will appear in the eigenvectors corresponding to the zero eigenvalue. So we do this for all the eigenvalues till the largest eigenvalue. So for the largest eigenvalue, the eigenvector is called the principal eigenvector. And the cross hubs that we get from those vectors are called the principal cross hubs. So basically, those principal cross hubs are might be different from the cross hubs we get from the zero eigenvalue, and that they are hubs, but they actually hubs between very connected clusters, potentially very connected clusters. So in fact, this is there's a spectrum. I mean, it's a very spectral in the sense that what you get at the zero eigenvalue might be hubs between non-connected regions, and as you go further on the spectrum, you get hubs that connect more connected regions that we get till the maximum eigenvalue, and then there you get the one that is a hub between highly connected regions. And this one we consider as the one that is responsible of the structure of your space. Okay. In the case of the airports I talked about, that will give you the answer. That will give you the airports that allows you to connect the most efficiently from one airline to another. So, and this is an example of, so this is exactly the result from this simple bicomplex. So this is the matrix corresponding to the cross-Laplacians. So this is the top Laplacian, and this is the bottom one. And these are the eigenvectors corresponding to the zero eigenvalue. So you see, we get, it gives you the two cross hubs. And this is the eigenvector corresponding to the largest eigenvalue of the top. So now you just get one hub, so V1, because this is, and you can see here, V1 indeed allows communication between this connected structure and the other connected structure. See, so it allows communication, intercommunications between two different areas, two fully connected area, actually subnetwork of the top layer. So this is what is represented by this graph, and we do the same for the bottom cross-Laplacian. Again, you just change the view. So in fact, in some sense, it's a way to see how the structure from one layer controls the emergence of some topological layer, some topological structures in the other layer. Okay. And that also translates into interdependence of the topological structures of both network layers. So now, because we are doing the same analysis for every eigenvalue, and because for every eigenvalue you get new, potentially new hubs, you can apply the same notion of persistency I talked about for persistent homology to look at the hubs that persist or that come up the most often during this process. So meaning that you are looking at the hubs that are hubs between separated regions, but at the same time, hubs between fully connected areas. There is a difference between that, okay? Because they can be both. I mean, it can be, let's say, 100 fully connected networks. You still have hub between separated networks. It's different between a hub that connects 100 non-connected areas. So both of them will be seen by the zero eigenvalue, but only one of them will be seen by the maximum eigenvalue. So only one of them will persist through the across the spectrum. So we represent the persistency by horizontal bars. So this is the spectral persistent bars of cross hubs. For example, here at 0, we have two hubs. At 1 something, we have other hubs, but they don't appear on other eigenvalues, and only the first one appear for the on the maximum eigenvalue. We do the same for the bottom layer. So we apply this technique on structure function, resting state fMRI data from HCP, and we applied on separately on both hemispheres, just to test the method. And so the color code here actually corresponds to the subnetworks. So I think the purple corresponds to the frontoparietal, the gray is default mode, and the yellow is visual network. And the blue one, I think it's the auditive network. So look at what happens on the maximum eigenvalue of only two regions, two cross-hub. So, okay, again, this is the partial correlations mapped on the anatomy. So now what you get are functional hubs on the anatomy. So, and when you look at the whole persistence bars, you see how hubs emerge, how functional hubs emerge on the underlying anatomy. The more they persist, the stronger they are, or the more central they are, functionally speaking. Okay, so what we saw is that we don't have the exact regions from left and right on the maximum and on the second. However, when we look at the last four eigenvalues on both sides, we get almost the same regions. So we have a symmetry between the left and right at the maximum eigenvalues. Not the maximum eigenvalue, but the last eigenvalues, actually. So this is the left, and this is the right. So you see you have almost exactly the same. There's no default mode region here, but we have the exact same regions from the frontoparietal and visual and auditive mode. So there's perfect symmetry between, in terms of functional hubs. So yeah, I mean, this is not a new result, but it proves that the method actually works. Now, we need to, the interpretation is not straightforward, of course, especially, so it's easier when you look on this way, I mean, from function to anatomy, but not from anatomy to function. What a hub, what would cross-hub means when you look at the function through the anatomy, by example. It would still be an anatomical hub, but what can you say in terms of function in that case? Yeah, so it's again, this is an ongoing work, but the method works. Now it's a matter of it's a question of interpretation that we need. So I think I will stop here, not to.
Idea of
Yes.
interpretation.
Yeah.
And I've been thinking about this, like, since you started talking about this. There's a new paper, I don't know if you've seen it, it's by John Bull. So it was just published in PNAS.
Mm-hmm.
And the data is available, and I think that that data is going to be very useful. So in this paper, so first of all, they're showing that the connectivity, and they're following up on something that Peter Banatinian is not on connectivity, but on the board response, that the board response is present in all of the brain. That the reason why they're finding these networks is that the hemodynamic modeling is not accurate, that we find, sometimes areas that are more connected than not connected is because it's tied to sort of assumptions about hemodynamic response. So in this data, they have fMRI in 5 different modalities. Yes, I'm talking about the John Gore's PNAS paper. So they have resting state, they have visual stimulation, they have left-right motor and left-right sensory stimulation together with DTI. And they're in fact showing that the connections in response to task within the DTI are also, regionally specific, although these connections exist everywhere, but then they're regionally specific. So I think that data set might be very useful in terms of finding the homologies between, say, left-right hemisphere in doing something that is very simple finger tapping motor test, but then at the same time is going to allow to sort of look at the differences in holes between, say, if you're looking at the data that is resting state versus the one that is motor. So that would be, I think that would be really cool to look into this data.
Yes.
And it has two modalities, so it allows you to do this.
To map both modalities.
Yeah, I'm happy to send you whatever.
Yeah, thank you. Thanks. Yes,
But it's really, that would be cool to look into it.
Yeah. Please send me the link and we'll look at. Yes.
First of all, thank you very much for your informative, nice presentation. I have a practical question. For example, let me just focus what we want to study, because this is the neuroscience concept. So we want, for example, if we consider functional and anatomical two networks in a specific set of nodes, okay? For functional connections, we can measure it and it's, specified. For anatomical connections, also we have measurements, but how we can measure between connections, how we can determine the value of connections between two networks, functional and
how do you connect both of them?
The nodes, the two nodes from two different layers.
Okay, so in this example, we didn't put any connections between them. We actually considered an empty graph and put the function in between. But of course, you can define yourself. It depends on the use case. You can define your own connections. You can say, okay, I want to see for example, correlation, so functional connectivity, and using defined by some, let's say, correlation, and have anatomy, and I put in between another functional connectivity defined from other method, let's say entropy, for example, or partial correlation.
But the nature of
it depends on what you want to do.
So it's
yes, it depends on what you do. In that case, for example, you would want to see how close are two types of functional connectivity, and how both map on anatomy, for example. So it really depends on what you want. There is no, I guess what I'm saying is that there's no canonical way to define the interlayer connections. It's all depend on your data. It all depends on the question you want to address. And that's it.
Because also the definition is very important.
Yes, of course. Yes.
The concept of multi-layer.
No. The concept itself is a general concept.
Yes.
You cannot say, okay, this is a definition of interlayer connections. No. It's intrinsic.
No, I mean
over data.
For
example, for the C elegans, this elegant case is quite interesting because this is in the neuronal scale. In that case, you have more connections to consider. For example, you can consider the synaptic and the gap junctions, and let's say monoamine connections between both layers. That's an interesting case.
For that example, is
Yes.
well defined. But
Yes.
for
for macro scale,
anatomical is
for large scale, it's not straightforward. It's not obvious to find the interlayer connections, and that's why we chose not to do for now not to go in that direction, but to project one layer into the other and look at one layer through that network, and vice versa. Which is also an interesting question, because in that case, you really, instead of comparing both network with the statistical traditional tools you have, meaning you study each one separately, now we just project one on into the other and study the whole structure.
And another question, how do you, because maybe I missed this part, how do you introduce the concept of dynamics in your theory?
Okay, so we haven't looked at that, but there's no reason not to consider dynamics here. As long, I mean, you have Laplacians here, operators. You also have adjacency matrices, but now the question is, what kind of, let's say, diffusions you have in the case of this L00, for example. The L00 actually encodes, so it sees cross triangles, and where there's no cross triangle. Okay, so what does that mean? So what is the diffusion between that follows cross triangles, for example? What does that mean? So that means something that is in the edges or the cross edges, and then that goes back to the other layer. So I honestly don't know of any for now. Yeah, we need some concrete examples of dynamics that follows that kind of path. But mathematically speaking, it makes sense. But concretely, that's another question. Yes.
And let me mention this one. When we talk about when we talk about diffusion process, for example, because in the case of my project, we talk about the diffusion of topology. Okay, so the diffusion is only consider a specific kind of a species. Okay, but in your case, if you want to consider diffusion, for example, in two layers, and one diffusion is related to, for example, the functional, and another diffusion, for example, to topology.
Yes.
And then, you have, because the nature of the links in this case different.
Of course.
And then combining the concept of diffusion.
Yes, that's why I'm saying that it's it's not obvious to interpret this kind of diffusion in this case in real data. You need to have actual data that incorporates this whole idea of diffusion in a very natural way. But yeah, in the case of functions, I don't see any way to what is a functional diffusion on the functional connectivity. What does that mean? It would mean just information that passes, I don't know, or synchronization. So that's something like that. But I
and this is my last question. Just This is the last one, please. It's important.
But sorry, but it could be, for example, in the case of the in the neuronal scale, if you have two layers, structural layers, and some other, it could be something like a diffusion of some substance that transforms into other substance that follows the new substance, that new molecules that can follow other path. Let's say something that's maybe concern the genes, and then the genes passes through the other connections to the same network, and then follow the other. That can be an example of cross diffusion.
Your theory is very interesting, but it needs a lot of, I mean, interpretation.
Yes.
And okay, just one, just
Yes.
Thanks for the nice talk. So, do you think it will be more easier to interpret this cross layer edge? For instance, instead of using functional as one layer, anatomical as another layer, like we can use functional fMRI and like say astrocyte network. So in that case, maybe they're all functional connection.
Yes.
So the cross layer edges may be more interpretable.
Yes. Yes. I think that was Obaid used multilayer. He didn't use this cross stuff, but he considered astrocytes as one layer and the neuronal network as another layer. And yes. I think he's considered like one neuron for one astrocyte, right? One astrocyte, one astrocyte. So there's nothing in between, and no way to. That's
One more question, last question. Yeah. So we talked about using anatomical like fiber tracks and guide like as an architecture for the connectivity. Here you already have the connectome for different modalities, and your tool is to kind of analyze the graph, like extract some pictures from the graph, considering the interaction between them. So instead, we have like anatomical structure already there, the connectome already analyzed. By using this inter-layer connection or more information guide the estimation of functional connection. I'm not sure if you want to brainstorm more like information instead of only looking the fiber flags and giving a weight, let's say.
So the question is,
is kind of a pen can help understanding and map it and the another so from the function to the anatomy. One is if you have data function of the data, if you would like to look at the function of the data through the anatomy, one way to statistics is to project the data from the functional data on the space stage generated by the anatomy. So in this space, the anatomy project is space. What I think that's project on subspace, and we don't have anything in subspace, how we got it. And in general, statistics, you to look at the national between different space, project this space. If you like one space of other space, so here are link is mass function. This is a how this function this let's say this what I can do. So the result so this is so where we detect then this is the function of is different from the help from the function as usual. At this stage, I don't. They're not the same. Yeah, they're not the same. I just think I only told you this way, but I also have the other way around. So we change the point of view of work based on this, and it's not only an anatomy, it's a function explained by the anatomy. So maybe this is like adding value of now how we can learn from the function of the anatomy both together. But I think that we can go more deeply than that. But this is about the question.
So how do you look for the dynamics in these hubs when you're projecting? So you have anatomy and then you have function. And let's assume that the function is not something, let's assume that the function is not entirely tied to the anatomy and that it's something that is moving, right? So say waves are traveling, the neurotransmitters are traveling. So how would you be mapping the time into this? Or is it possible for example?
Yeah, I think that's also that is related to a question Arsalan was asking about how do we integrate the dynamic and this formalism. This formalism, mathematically speaking, it is straightforward. But again, the question would be, yeah, it is doable using this Laplacian, but again, we really need to interpret this kind of result even for static data before we even think about the dynamics in that case. But yeah, so I don't have an answer for that right now. Yeah. I don't know if
just to relay what Arsalan mentioned, because I think dynamic is very important. So how the geometry change over time. What are the invariants? So this question has been asked by Victor Girsa to Arsalan, saying, okay, oh, your space is fixed, but the concentration of the tau protein, the species, vary over time. This is the differential equations. And one of the response of Arsalan is the fact that he looked at the Laplacian operator L by xn, x power n. And x power n, x is the concentration. Power n is the nonlinear diffusion. And so the interpretation of this by Victor is to say N doesn't change. But this is bizarre because he showed variation over space of this concentration over time. And the response comes with, oh, don't consider L by x of power n, but L by x of power n minus 1, and we multiply by x. So if you consider L by x minus n minus 1, we show that L is now guided by x n minus 1. So it changes over time. And he said, yes, I see it now, because we now, but it's a particular case where we modulate the space by the concentration, which concentration change over time. So we can consider a more general point of view here, considering a different dynamic system, and this is part of the response for Arsalan, where for some times window, we define exactly as the persistent homology that we did. What persistent homology took coefficients, defined a network of relationship, then look at this this coefficient over, I talk about the significance in functional connectivity, 1%, 2%, 5%. Now took the dynamic, and rather than 1%, I took in sometimes window the relation. So I have one graph at one time window. I have a second graph at a second time window, and I can plot them together. So I come with this point of view of multi-graph, but each one inform about what's happening at some time window. We can also look at the relationship between one node, how the nodes evolve individually. So we can make a link between two levels of the graph. It's not at some time window I am related this way, but between time t and time t plus 1, maybe I change my relationship with the nodes. So I can code for this dynamic relationship between. So what in this case I encode.
Yes, in that case, yes.
Within the relationship between the graph. And I can analyze the whole spatial, temporal evolution.
If the question is, I mean, you have function that evolves over time, in that case, you just consider multi-layer networks. For each time window, you consider a layer, and then you map into the anatomical network, which is fixed. And also you can map one type T1 to T0, etc., and look at how how these cross hubs evolve over time. In that case,
I have something in mind, like Grange causality. So this is something that I can put between two nodes. How at time k plus 1 can be predicted by what's happened before k. So this is a prediction 1-to-1. Okay, so this code for this Grange causality between two slides, and in one space, what is the coherence between all the connectivity that I observe? which is another question. This is a space where I evolve from the connectivity point of view. And when I move from one step to another step, I consider the timing as predictor, how I can predict for this part. So if I encode between Granger causality and connectivity, what is the geometry behind this? And his tools can respond to this kind of thing. So there is a lot to say, a lot to test. We never ask this question yet.
Yes,
but
it's a consequence of this.
Yeah.
Somehow, so there is a very nice paper I saw published in Nature or Nature Communications. I don't know, but called "The Shape of Memory." So they studied temporal networks with the same idea, using multilayer networks.
Question?
And they studied the topology of each network at each layer, and that give them actually what they call the shape of memory. So somehow, does the memory have shape? So in that case, yes, does dynamic have shape? So that's cross-homology would address this question.
Yeah.
So thanks.
I think there is a lot to do, because you talk about memory, and I am thinking about how we can, in the dynamic processes, if I can encode between two layers, like a transition matrix, like a Markov process. I have a dynamic process, and this transition matrix help understanding the relationship between different steps of time, because this is what the Markov process says. I move from t to t plus 1, and I have this transition in a simple case. So how I can encode for this information between the different layers? And is this shape the geometry or the relationship between the different layers? Another fundamental question that we have never addressed, but at this stage, we have the tools to do it.
I'm thinking really far ahead. This could have, this could have clinical implications, right? Because if you want to say, for instance, cures mental illness, right? When you have this, you're assuming that there is something deficient with the structure and the chemistry and the two of them over time. So how would the topography of a network change with treatment over time? You use the excellent word of the shape of memory. And what is the shape of recovery?
Yeah, yes.
And then, what do we arrive at?
Yeah, you look at the network at different stages.
Like we map this state of functions, whatever function space.
Yes.
Connections, and then we see
how these cross-hubs move over time. Yes. Yeah. Yes. That's Yes, that's an interesting
I have another question, which is
indeed.
I think that Arsalan does not ask for this question, but I ask for in the own of Arsalan, because he is dealing with connectivity. But your graph are not. There is no orientation in your graph.
No.
Okay. Is there any possibility to extend this a non-oriented graph to an oriented graph and look at this topology within oriented graph? Because what Arsalan is doing is
oriented
DAG.
There is, but it will be the same for monolayer, for the monolayer case. That in that case, because it's all about your definition of simplices. So if let's go back to the case of monolayer graph, so simplicial complex. So if your graph is oriented, so how do you define a triangle? So the most natural way to define a triangle would be something that respects the orientation. So A goes to B, B goes to C, and C goes to A. But the problem with that is too much restriction, because of course, the problem is that those kind of triangles can be really, really rare in real data. Okay. In some data, many data sets, you don't have such kind of configurations. For example, one of the kind of data sets that are used in complex networks are the citation networks, for example. It's a very concrete example, citations. You say that, well, article A maps to article B if it has cited article B. In that case, you would never have that kind of triangles, because A cited B, B cited C, C, so A cited, yes, there's no way for C to cite A, because that means that A is in the newest article, right? If A has cited B, B has cited C, so it's impossible that C cited A, unless it's an addendum. So, okay, so there's no way to do that. So you never have that kind of triangles. So this kind of orientation would not make sense. So yeah, so you either forget the orientation if you want to use simplicial homology or adapt your data in a way that it makes sense for your questions.
Or to extend the homology.
Or to extend, yes, or to extend this thing, but yes, the homology and to get something else. And that's what from this question, in fact, we've been also developing some new way to formalize, to do some kind of homology for oriented graph. That makes sense. But that theory will, is really looking at the flow. It's a homology of the flow over the graph rather than the orientation. So it takes into account the orientation, but the most important thing that we are addressing is how do we quantify the flow, the inflow and the outflow of your graph, meaning that you're looking at one node and you see how the information flows into that node and how it flows out of that node. So that doesn't require to have very direct triangles or else.
Okay, this is just to open doors between discussion between Akbar and Elka in the future.
Yeah. So yeah, we'll talk about this after when the paper will have been published.
Okay.
But,
in